So I have the broth or the dilutant, so the thing that I'm going to dilute the probiotic in. It's already sterilized, as you can see the black lines on the sterilization tape. It's letting me know that the giant pressure cooker, the autoclave, is working as it should, and so my things are sterilized. And what I'm going to do is dilute the 1 grams of the probiotic into one of this tube, mix it, shake it, then take 1 ml from here, put it into this, pipe it up and down to mix. 1 ml from here into this, pipe it up and down to mix. 1 ml from here into this, pipe it up and down to mix. 1 ml from here into this, pipe it up and down to mix. Once that's done, I'll take 0.1 ml or 100 microliter from each of these and put them onto agar plates. And that is what the cells, the yeast cells from the probiotic, will grow on. And after a few days, I'll count it. I'll do a calculation to see if it actually contains the 5 billion cells that the probiotic says it contains. So come along. So one capsule contains 0.53 grams, and I start by filling the 0.53 grams into a Falcon tube. So that's my original sample. I shake it very well, and now I'm going through the serial dilution stage where, as I said before, I take 1 ml from my original tube. I put it in my first tube that contains 4 ml of the dilutant, so the solution in which I'm diluting the probiotic. And before I put it into the next tube, I flame, and that's why my hand keeps going to that thing at the side. And I keep doing that until I'm complete. So you can see the gradation from very dark to light in the test tubes. So from most concentrated to least concentrated. Here I'm setting up the plates for the plating part. So this is a part that will give me the opportunity to count the number of cells in my probiotics. So I'm plating out 5 of the dilution. So I did 5 of them, and I'm doing it in triplicate. So each dilution, so the most concentrated to the least concentrated. So 10 to the 1, 10 to the 10, 10 to the 100, 10 to the 1,000, 10 to the 10,000, and so forth. I'm going to pipette. I'm going to take 0.1 ml. That's what I'm doing, and put it on the plate that represents the dilution, so the test tube. Then I'm flaming my hockey stick to sterilizing it, cool it, and then spread the zero point 0.1 ml that I put on the plate. Let it air dry before I seal it by wrapping the plate and the lid in parafilm, and then I'll put it under the right conditions of light and temperature to grow for maybe 5 days or 3 days. I'll check it when I'm in the lab to see if there's growth on the plate. So, to provide some context, I want to have between 30 to 300, or it could be 20 to 200 individual colonies or cells growing on the plate. And so that is why I did the dilution series. At the end, I will select one of the dilutions that have that range of cells in it that looks like I can count it. And then I'll count those, do a calculation to figure out how many cells are actually in my probiotic. So that is the whole serial dilution and plating. Pausing to show you what flaming the hockey stick or the L-shaped glass rod looks like. So I swirl it in ethanol, about 95%. I put it into the back incinerator so it doesn't have an open flame to disrupt the HEPA filter. And then I let it cool. and that sterilizes the spreader that I use to spread the probiotic onto the plate. So it's a series of aseptic techniques, flaming, sterilizing in between. So once I'm done, I do not leave the plates to incubate for the probiotic to grow just so. I actually seal it with paraffin, which is a stretchy piece of plastic, and that will prevent contaminants, so things that I don't want to grow from entering the plates with the agar that has the probiotic cells that I want to grow. So I'm sealing it with a paraffin, and once that's done, I put it in the incubator. That's what the plate look like with the cells on it, like when I just put it on it. And then we'll come back in a few days to see if we actually see growth. So right now they kind of look empty. So this is a few days after, and well, there's condensation here, but you can see it's smeared, lot of growth. So I couldn't count that one, and they all seem to have a lot of growth. Yeah, even the most diluted one has a lot of growth. That would be difficult to count. So I'll have to redo them and increase the dilution series. So I stopped at 10 to the 5 dilution. So I'm going to go to 10 to maybe the 10 and see if I'll have growth on the lowest ones. I repeated the serial dilution and plating, so you can see in this case I have 12 plus the first one 13, and for the very first one that you're seeing, the thicker tube, which is a 50 ml, I actually diluted the probiotic in the capsule in a larger volume of water and then do the serial dilution and plating. But as you can see, you see the most concentration where it looks cloudy and more brownish to almost looking like there's nothing in the tubes as you get towards the end. So I'm going to come back in a few days after they've been growing to do the calculation to actually see, do we really have 5 billion cells in my probiotic? So stick around for that. That will be the final video in this series. Bye.